So the top strand right here, that is our original DNA template. Okay, so we have unwound it. We are reading, right, from 3' to 5'. We are synthesizing. What does synthesizing mean?
Changing? Like copying? No, that's right.
Making, right? We are synthesizing from 5' to 3'. Okay, so what do? We replace what letter with what letter when we go from DNA to RNA?
T and U.
Right, T is replaced with U, right? So in DNA, it's G, C, A, and T. In RNA, it's GCat and GCow. In RNA, it's G, C, A, U. Okay? So, here we go. We are replicating, right? What do we read? What do we read in order to make an amino acid or a protein?
Numbers. RNA. Letters.
We read the RNA. How many bases do we read at a time?
3.
3. And what is that called, boys, over here?
Oh, it's
Base triplets or codons. Okay? So here's 3. Here's, whoops. No. Ah! Oh no, where did it go? We'll get it back, it's okay. Okay, so we have AUG, ACC, CAC, GAU, GGG, UGA. Okay, UGA is our stop codon, so we don't code for that. If I had a missense mutation, a missense means I change a letter for some way, shape, or form, right? Like I read the code and I'm dyslexic or I make a mistake, right? I'm not changing anything except for a letter. So here is our missense. Correct. Okay, and we also have DNA polymerase, right? There's a lot of enzymes that go through and they read that code. That DNA polymerase, it goes through and it reads the code as it's sealing it up. Okay, so that DNA polymerase is supposed to catch any issues because that RNA primer comes in, reads the strand. Okay, the primase comes in, it brings you the bases that it needs. The ligase goes and locks it all up, and then the DNA polymerase is that last final piece, right? That goes through and it reads it up and it zips it all together. If we're just doing for the RNA strand, okay, you have the polymerase that comes through and it reads that strand and makes sure that it fits. Okay, so hopefully it'll catch the errors and it'll, no, cut this out, redo it. But if it doesn't catch the error, it's called a missense mutation. And we talked about this yesterday a little bit. Some of these codes are similar enough that as long as you don't mess with the first 2 letters, that third letter doesn't really matter, right? You can change it and it's not going to change the type of amino acid that it's going to code for. However, this is the first letter in that codon. Okay, so we change from a THR amino acid to a SER amino acid. Is that going to change what my protein looks like? Yeah. Right? Okay, so that's a missense mutation, meaning that we misread a letter and we alter that letter. Okay, we have a frame shift mutation, meaning that we actually shift our frame over. Right? So we take the rest of this. So basically we have deleted this, right? There should be an A, G, 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 T, C, but we delete that A. Right? Instead of reading it, we just omit it altogether, and then what does that do?
Everything there is one over to the left.
Right, everything after it is affected, right? And then look, we get here and we have just 2 bases, so we don't even have a codon. So, with a missense mutation, how many amino acids are affected? Up to 1. Could it be not affected at all?
Yes.
Yes. Okay. With the frame shift mutation, which amino acids are affected?
All of them after that mutation.
That mutation and everything thereafter. Here's another example if we're actually looking at the chromosomes themselves. How many pairs of chromosomes do humans have? 23. We have 22 homologous pairs and 1 pair of sex chromosomes, right? So we have 23 pairs total. So if we're talking about a mutation, we might have, boys, okay, we might have a deletion, meaning that this piece is actually deleted, so we end up with a shorter section. Boys, please. We might have a duplication, meaning it might read AAA, and it might put it in twice.
So basic accounting errors?
We might have an inversion, meaning that it might say AUG, but it's going to be dyslexic and read it the other way, and it's going to say GUA. We can have an insertion, meaning that we're going to take this piece and we are actually going to insert it into DNA. So we might have another RNA strand and 2 RNA strands being read at the same time. We might take...
Would you replace it with that one, though?
What do you mean?
Like, since you're inserting one, would you just be adding on to it, or would you be, like, replacing part of it?
You would be inserting a new piece.
Straight now.
Okay, and then there's what's called translocation, meaning, and this is how we get genetic variation, and translocation can be actually a really good thing. So we have a chromosome from our mom and a chromosome from our dad, correct? So when we actually have mitosis occur, what happens is that DNA duplicates and it's really close. So we have 2 copies from mom and 2 copies from dad, and they're really close together. Well, what can happen is part of that DNA can cross over, and then I'll have part of my dad's DNA and part of my mom's DNA on that one strand of DNA. So when I separate that, right, if we're talking about meiosis, okay, we're looking at creating sex cells. So if I have a translocation, meaning I have part of my dad's genes and part of my mom's genes on one big long strand of DNA, and meiosis occurs, I could have one cell that actually gets part of my dad's DNA and part of my mom's DNA.
Wouldn't that be you, though?
What?
Wouldn't that be you, since that's a mix of both?
Well, I'm sorry. I'm going to get part of my grandma and part of my grandpa.
Or like,
Right.
Basically,
Is that normal?
say,
That would be you.
Right. Well, yeah, because you have half from your mom and half from your dad to create you. But when your mom's like egg cell or when your dad's sperm cell goes through meiosis, which means it splits the DNA in half, right? It'll take a piece from their mom and a piece from their dad, and it'll make it one big strand of DNA. So instead of no, but instead of only getting one section from your grandpa or one section from your grandma, you might get a mix of both of those, right?
Right, which would be why we have...
Which increases genetic variation.
Or, wouldn't that also be complained to be two-colored...
Two-colored eyes? It could be. Or that could just be a missense in your protein.
Right.
Okay, so does that make sense as far as mutations go? So let's look in here.
I'm going to say basic accounting errors.
Oops. So here is an example. I want you to look at this. These are types of mutations. So if I'm looking at this, these are the types of mutations that we have. So as, we don't code for as. We start on the. Okay, so this sentence should read, the man saw the dog hit the can. End, right? That end is cuts off, right? If I have a point mutation or a missense, we are replacing a letter. So the man saw the dot hit the can. If I have a deletion, I can say the man saw the hit the can.
I think he's illiterate.
Right? If I have an insertion, I can say the man saw the fat dog hit the can. Or if I have a frame shift, and frame shift is the one that really doesn't make sense, right? That's the one that affects everything else. The man saw the it, heck, and it's, right? Doesn't even make sense. Right. This is my cell phone is losing service. I'm recording right now, and I guess you can't really see it, but down here there's like a little red record button, and I'm talking, and I'm writing, maybe, and then I go, oh shoot, somebody just said the F-bomb in the back of my classroom, and I don't want that on my YouTube video,